And our next speaker to end this little portion of the session is Chaitan Khosla. Those of you who know Chaitan's work might think of him as a natural products chemical biologist, but he has another hat on today, and he's been making important contributions to celiac disease, and that's what he's going to tell us about today. So go ahead and take it away, Chaitan.
Thank you, Carolyn. Let me also start.
Except for I'm Laura. I'll just.
This is like the. I wanted to thank Carolyn for having introduced Bob's contributions to the field. As a chemical engineer, I was thinking about how much of an impact Bob has had on chemistry, biology, medicine. He has more bandwidth and vision across these disciplines than probably anybody I know, maybe everybody I know. And so this is a really big honor to have a chance to share this this stage with Bob and tell you about the work that we've been doing on celiac disease. Let me also point out that I have a couple of conflicts relative to this talk that I need to disclose. Let me start by introducing to you what celiac disease is for the sake of bringing everybody on the same page. Most of you probably have heard about celiac disease. It's a gluten-induced inflammatory disease of the small bowel. It also affects other organs. So for example, what you see in the adult patient to the right is a pruritic rash, which is a very common manifestation of untreated celiac disease. It's an autoimmune disease like your favorite autoimmune diseases, whether you think of type 1 diabetes or multiple sclerosis or rheumatoid arthritis, in the sense that it has all the hallmarks of T cell and B cell induced inflammation. And it's a widespread disease. It's about 1 out of 100 to 200 of us have this disease, but notwithstanding the fact that it's been over 60, 70 years since the disease was recognized, there has really been no efficacious treatment outside of strict dietary exclusion of gluten on a lifelong basis. To give you a sense of scale, 20 parts per million of gluten in a diet is about the equivalent of the amount of gluten that 1 crouton would contribute to your diet. So this is a significant challenge out there for a patient. Around the time when we started working on this problem about 20 years ago, this slide summarizes what was known about the pathogenesis of celiac disease. It was well known that gluten was in our diet was the primary trigger of gluten proteins are the primary trigger of celiac disease. And it was pretty well known by then that this is a T cell driven disease in the sense that the gut of a patient has gamma interferon producing inflammatory T cells that are activated when a patient consumes gluten, giving rise to all of the sequelae associated with the disease. And there were a couple of key insights that were starting to emerge around then. The first one being that for gluten to activate T cells, you needed to proteolyze gluten into peptides. And some of these peptides, which were proline and glutamine rich, were good ligands for these disease-specific T cells. These disease-specific T cells were activated by these peptides bound to a particular major histocompatibility complex protein called HLA-DQ2, which recognizes these gluten antigens and presents them to the T cells by well-established mechanisms. But what was particularly interesting was that in order to be able to recognize these antigens, they needed to be first modified by an enzyme in the gut, a human enzyme in the gut called Transglutaminase II, or TG2 for short. And what this enzyme did was recognized in a regioselective manner certain glutamines in these peptides, converted them into glutamic acids, and these modified peptides were then presented on HLA-DQ2. And somehow these T cells were also driving the antibody-producing B cells associated with this disease. So this was more or less what was known about 20 years ago, was starting to be, recognized. And coming at it from a chemical perspective, the obvious questions that any of us would ask were the questions that our lab asked around then and continues to ask, which is, what is special about the three key molecules that one sees in this pathogenic scheme? What is special about dietary gluten? What is special about HLA-DQ2? The human HLA repertoire consists of many different MHCs. Why do 95+% of all celiacs have HLA-DQ2? And what is special about this particular isozyme of the transglutaminase family of enzymes that leads to this disease? And I would say most of what I have to present today is motivated by these three mechanistic questions. So the answer to why gluten, among all of the proteins that we consume in the human diet, why is gluten special, is now pretty well understood. If you take what I'm showing you over here are the amino acid sequences of on the order of 1,000 gluten proteins that exist in a typical bag of flour that you may buy at the grocery store. You can see that these are proline glutamine rich sequences. And what you're seeing in red in each of these two sequences are stretches about 25 to 35 residues that are virtually untouchable by any gastrointestinal protease in mammals. So essentially, these sequences in red are the equivalent of sand, while the rest of the sequences of these proteins are what provide the nutrition. Ultimately, over time, these peptides do get degraded, perhaps catalyzed by processes that are promoted by the bacteria in our gut. But these peptides are not just metastable entities that are derived from gluten. They also comprise all of the major antigens that are recognized by the T cells in celiac patients. So in that sense, celiac disease is somewhat unusual in the sense that it is driven by a dietary protein that is extremely nutritious to all of us. But to about 1% of us, it generates these antigens that are highly polyvalent. I think our session chair has had a rich history of illustrating the power of polyvalency in various cell surface processes. This is a beautiful example. So this particular 33 residue peptide shown in the sequence to the left has 7 epitopes, all of which are extremely potent epitopes that are recognized by human HLA-DQ2 after they are modified by this transglutaminase enzyme that I referred to you. And this protein, the sequence on the right, has 6 epitopes that are all recognized by HLA-DQ2. So these are incredibly potent antigens that are just not digestible trivially in the human gut. And so that led various students in my lab several years ago to think that we could accelerate when one might be able to treat this disease by accelerating what is the rate limiting step in gluten digestion in the gut lumen. For most other proteins that we consume as a dietary source of nutrition, these initial steps of breakdown of the dietary protein are slow, and then the breakdown from these peptides to amino acids and dipeptides that are absorbed into our bloodstream is extremely fast in the gut. And so there's really not much buildup of these peptides. And so, but in gluten, you have a different story because some of these peptides that I showed you in the previous slide build up in a very significant way. And so the thinking that Lu Shan and Michael Bethune and John Gass in my lab over the years developed was that you might be able to engineer proteases that have the right kind of selectivity, that they can recognize these epitopes in the mixture of food that one consumes and alleviate this rate limiting step in the breakdown of gluten proteins that we consume. That hypothesis led us to engineer a protease that today its non-proprietary name is ladiglutenase. And this protein has been validated in a number of clinical trials as being efficacious. The first proof of clinical efficacy was derived in a phase 2 randomized controlled trial in Finland, whose data I'm showing you over here. In this study, one was essentially using, it was a relatively small study on about 40 patients, where who were all challenged who were more or less in remission because they had managed to exclude as much gluten from their diet for many months. And they were then challenged with a relatively small amount of gluten. That's about the equivalent of a slice of bread, 2 grams of gluten per day, for about 6 weeks. And the key measurement that was made was, I don't have time to go into the details, but this is the sort of histopathological gold standard of celiac disease. If you take, it's measured, it's a quantitative measurement that's made on biopsies of celiac patients. And you start with a fairly high number because these patients are generally in remission. And over a period of 6 weeks, when these patients are challenged with gluten, this number drops. And what you can see is that while the patients who were challenged with gluten in the presence of placebo, this number dropped very significantly, the number was relatively stable in patients who were treated with ladiglutenase, suggesting that this drug did have clinical efficacy. However, the FDA does not consider histopathological measurements as a valid measurement for approval. And so many years and many millions of dollars later, a clinically relevant tool that correlates histopathology and other gold standard markers, objective markers, to clinically relevant indices like abdominal pain and tiredness. So a tool was developed, a patient-reported outcome tool was developed. Since there's never been a drug developed for celiac disease, one had to essentially develop a tool to measure drug efficacy from scratch. And so at the recommendation of the FDA, a tool was developed. I'm just showing you 2 examples of metrics that were used in the tool. And the study that was done in Finland was essentially repeated at the Mayo Clinic with an additional 40 to 50 patients. And you can see now the histopathological result, which was reproduced from Finland to Mayo, was also shown to have clinical relevance in terms of symptoms experienced by patients. But what I particularly want to point out, and I'll come back to this if time permits, is that in addition to being able to see effects on symptoms, one could also measure that there were urinary peptides that were derived from gluten that were significantly higher in patients who were treated with placebo. And that probably is the best metric of the clinical efficacy of the drug. If gluten were alcohol and you were developing a drug against alcoholism, the FDA would approve a drug based on your ability to show that the blood alcohol levels in an alcoholic go down when you treat somebody with a drug. I'd like to suggest that gluten is at least as toxic to a celiac patient as alcohol is to an alcoholic. And so technically, this could be a very powerful marker. I also mentioned we were interested in the question why HLA-DQ2 and why transglutaminase II are important in this disease. And the answer to that question came from a crystal structure that Chu-Young Kim solved in our lab several years ago. This is an example where a picture is really worth 1,000 words because it illustrates exactly, for those of you who are familiar with HLA-D, class 2 MHC proteins, you know that they have these big grooves through which into which peptide antigens can bind. And much of the affinity that they derive for their peptide antigens comes from backbone hydrogen bonds. So this presents a challenge for proline-rich peptides in the backbone. And what you see in this crystal structure is that the prolines that are positioned in the gluten antigens are positioned exactly at the places where backbone amide bonding is not critical. So in some sense, there's a perfect match between this HLA within the human HLA repertoire and what exists in the wheat field in terms of where backbone hydrogen bonding is important. The answer to why transglutaminase II is important in the pathogenesis of the disease comes from the location of this glutamic acid residue that you're seeing at the P6 position over here. This glutamate didn't exist in the wheat field. It was generated by deamidation of a glutamine residue by TG2, and it's the critical salt bridge that gives HLA-DQ2 its specificity. So there's a unique lysine in human HLA-DQ2 that is absent in other HLAs, and this hydrogen bond is really critical. Now, as we started to appreciate what was important about HLA-DQ2 and TG2, what became also clear was that because DQ2 is a gain of function type of a target, it probably is an undruggable target in the context of this disease. However, TG2 is essentially just a cysteine protease-like enzyme, which operates on the side chain amide of protein rather than the backbone. And so perhaps one might be able to drug that. This is a problem where chemistry actually moved faster, considerably faster than biology. We needed to engineer tool molecules that could allow us to study the biology and pharmacology of TG2. But at the same time, because there was no animal model for celiac disease, there was a need in the field for a mouse model of this disease. Our role was to engineer the relevant molecules, starting from the X-ray crystal structure of TG2 bound to a gluten peptide mimetic. We were able to engineer a variety of small molecule tools, this being a particularly useful small molecule inhibitor of TG2 that had the right kind of selectivity for us to be able to take in vivo potency and selectivity. Another key molecule that had to be engineered was a probe to visualize TG2 activity in the gut of a mouse model, or perhaps someday even a human patient, because TG2 by itself is inactive in the healthy gut. It is somehow activated as a result of inflammation. And so for that, we engineered this biotinylated amine that reacts to glutamine residues in the course of the TG2 catalyzed deamidation. And so you end up with a biotin stuck to the substrate that TG2 modifies. Using this kind of a tool, we were able to help our collaborators, Bana Jabri's lab at the University of Chicago, who were in the process of engineering a mouse model of celiac disease, an effort that took them about a decade to do. And a key early result that was telling us that we were going in the right direction was when they had a model, a mouse, where when it was treated on a gluten-free diet, you could see that this probe doesn't stick to the small bowel, but in the presence of gluten introduced into the diet, if you inject this probe into the tail vein of the mouse, you see that it lights up. And when you co-inject the inhibitor, now you don't see the probe binding. So this tool was used to engineer further the mouse model to a point where one could actually do the non-clinical test of our hypothesis that if you block transglutaminase, you might be able to achieve clinical benefit in a celiac patient. And so essentially what is done over here is you take these reverse engineered mice and you treat them, challenge them with gluten, and then either you keep some of them on gluten or you treat them with gluten in the presence of a TG2 inhibitor and look how their guts behave. And what you can see is that the mice that are treated with gluten, in the absence of an inhibitor, they show strong levels of inflammation, whereas the mice that are treated with the inhibitor, there's a blockage. And so this compound was the basis for further engineering by a company that spun out of our labs called Citari Pharmaceuticals and was about 2 years ago acquired by GlaxoSmithKline. And so there is today a compound, that small molecule, that is undergoing clinical trials in celiac patients to be able to test the hypothesis that this non-clinical experiment suggests is worthy of testing. Let me just end by pointing out, and this might be a good place for me to again speak to the impact of Bob Langer's contributions to drug delivery. An important area that delivery tools have been quite impactful in is developing non-invasive approaches to be able to monitor diseases. So in addition to developing therapeutics, these delivery tools have also been impactful in developing markers. We've used them over the years. I don't have time to describe them, but there's one particular result that I alluded to that is worthy of emphasizing because it kind of violates every rule that we've learned about what drugs, how drugs, at least bioavailable drugs, work. I don't think any of us think of peptides as orally bioavailable molecules. Nonetheless, as I explained to you in my earlier slide, here's a 33 residue peptide that is consumed by us in the context of a protein that persists for long enough and is transferred, it is absorbed by the patient to be able to trigger an immunotoxic response. As we learned about this peptide, there was a company, Biometal, that developed an in-home test using this peptide to be able to, so for patients could test their food products for the presence of gluten. And people started to use these same tests to look for gluten in pee. Now, I'm not sure I would have predicted that this test would be useful over there, but it turned out it was an incredibly powerful tool. And so Brad Polanski and Nielsen Weng developed this human experiment in my lab where they essentially started challenging human patients with, as well as healthy volunteers with gluten, and looking at their urine over a day-long collection to see what they would see in an unbiased detection strategy. And what you see is quite remarkable. Our urine is full of gluten-derived peptides. And this is true for healthy as well as celiac patients. When we eat wheat, we see lots of wheat-derived peptides. When we eat barley, we see lots of barley-derived peptides. When we eat rye, we see a lot of rye-derived peptides. You don't see this with other non-gluten proteins. But what's particularly interesting is that the best markers, when you do an unbiased detection of what's in urine, the best markers turn out to be native innate immune markers, things that trigger innate immune responses in celiac patients. And so I think that opens a door for us to think about how our diet gives rise to macromolecules that travel all over our bodies, some of which do things that, who knows, some of are probably good, others are not so good. And hopefully tracing these molecules over the coming years will teach us something. I do want to acknowledge the people who've taught us everything about celiac disease. One of the benefits that we have derived from working on celiac disease is an opportunity to work with multiple generations of tertiary care clinicians, Gary Gray first and Nielsen Fernandez Becker at Stanford, as well as leading immunologists, Bana Jabri at the University of Chicago and Ludwig Solid in Oslo. Thanks again and congratulations, Bob.
Thanks, Chaitan. That was really beautiful work. And I'm gonna say, if anyone has questions, to just put them in the chat. I'll just ask a quick question, which is, do you know at all about the interplay between these peptides and what the microbiome might consume? So is there a component that you could look at to see whether or not there were certain microbes that might really enjoy these particular products?
Peptides?
Yeah, some of the intermediates that when you talk about, so there's the autoimmune response, I would guess, would influence the microbiome. And so I was just curious if you have any mechanistic link between what you've observed in your molecular analysis and the microbiome.
Are you thinking about post-translational modifications to these peptides, Laura, or just nutrition?
No, I'm just thinking they might act as nutrition sources for different microbes differently.
Absolutely.
And so then you might be like promoting the growth of microbes that you don't really want because they can really metabolize these, whereas other things can't.
That's a very important area to study. The challenge over there is what you need for that is tools to pick up your microbiota from the other end of the gut. Most of the microbiota that people sample is from the lower side. What you really need, the question you're asking, the most interesting target bacteria are in our upper gut, and we don't get to sample a lot of them by traditional methods.
Yeah, No, that is for
That's a
certain.
good point. Yes.
But I think now that you have a mouse model, you could actually use that to do some of those kinds of studies.
Yeah, it's a challenging animal. You got to talk to Pana Jabri about
Okay.
that.
Okay, does anyone else have any questions? There maybe sometime when I'll chat with you offline. We might have some tools that could be useful to you.
That would be very helpful. This is a tool-starved field. In fact, one of the things we're trying to do is build a network of investigators who could bring avant-garde tools. And so you couldn't say something that gets me more excited than what you just said.
Well, thanks, Chaitan. Thanks for showing us how you've been tackling this from a very different perspective than had been going on in the field.